Pseudo-organic life refers to complex, lifelike molecules or structures that are not the product of biological processes but instead arise from purely chemical, abiotic reactions. In permafrost environments, regions of permanently frozen ground, researchers have discovered intricate organic compounds and self-assembling structures that can mimic certain characteristics of living cells. These findings have opened up intriguing questions about the boundary between chemistry and biology and how life might originate under extreme conditions. For instance, in the icy depths of permafrost, chemical reactions driven by low temperatures, radiation, and mineral catalysts can lead to the formation of complex molecules that resemble biological macromolecules. These compounds can sometimes form vesicle-like structures, which are similar in appearance to cell membranes, suggesting a potential mechanism for the prebiotic organization of matter. How might these life-mimicking structures inform our understanding of the origin of life on Earth, or even on other planets? Studying pseudo-organic life in permafrost has significant implications for astrobiology. If similar abiotic processes can create lifelike compounds in Earth's frozen regions, it raises the possibility that analogous phenomena could occur on other icy worlds, such as Mars or the moons of Jupiter and Saturn. Some researchers are keen to determine the precise conditions that lead to these chemical self-assemblies and whether they might serve as stepping stones toward the emergence of true biological systems. Could these proto-like forms be the missing link between non-living chemistry and the dawn of life? Moreover, exploring these phenomena helps scientists distinguish between genuine signs of life and abiotic processes that merely resemble living systems. This is crucial when interpreting data from space missions, where identifying true biosignatures can be challenging. By understanding how pseudo-organic molecules form and behave in extreme environments, we gain a clearer picture of what to look for and what to rule out in our search for extraterrestrial life. In summary, the study of pseudo-organic life in permafrost is a fascinating area of research that blurs the line between chemistry and biology. It challenges us to reconsider our definitions of life and opens up exciting possibilities for discovering lifelike processes beyond our planet. What do you think are the most compelling implications of finding such complex, abiotic structures in frozen environments, and how might they change our search for life in the universe? Pseudo-organic life refers to complex, self-organizing molecules found in permafrost that exhibit some characteristics of living organisms despite being abiotic. These molecules are formed through non-biological chemical processes that mimic the patterns typically associated with life. In permafrost, extremely low temperatures and unique chemical conditions allow for the stabilization of these intricate molecular structures. Researchers study these systems to understand the boundary between living and non-living matter. Scientists have discovered patterns of carbon-rich molecules in Siberian permafrost that resemble the organized structures found in biological cells, even though they were formed through purely chemical reactions. It's like finding a beautifully arranged pattern in a frost-covered window. While the design appears organic and intricate, it is actually just the result of natural freezing and condensation. In permafrost, the combination of low temperatures and specific chemical precursors leads to the formation of complex molecules through abiotic processes. These molecules arise from reactions that occur slowly over thousands of years, allowing intricate structures to emerge. The stability provided by the cold environment prevents these molecules from degrading, making them persist in the frozen ground. This natural laboratory offers insights into how non-living matter can organize in ways that resemble life. Researchers have observed that certain organic compounds in Arctic permafrost can self-assemble into structured networks, similar to how polymers form, even in the absence of biological catalysts. It's like baking a slow-cooked stew in a winter cabin. The low temperature allows flavors to develop gradually, creating a complex dish even without constant supervision. Permafrost contains a diverse array of chemical compounds, some of which exhibit complex lifelike properties despite their inorganic origins. The unique geochemical conditions, such as high pressure and limited oxygen, contribute to these unusual molecular arrangements. Advanced analytical techniques reveal that these compounds can form self-organizing structures similar to those in living systems. Studying this complexity helps scientists explore the potential pathways for the emergence of lifelike systems in extreme environments. Analyses of permafrost samples have identified intricate carbon structures that, while non-biological, display network patterns reminiscent of cellular membranes. It's like finding a mosaic made from naturally occurring stones. The arrangement is beautiful and complex, even though it wasn't deliberately designed by an artist. Pseudo-organic molecules in permafrost can mimic several key features of biological life, such as self-assembly, self-replication, and structural organization. Although they are formed abiotically, these molecules may organize into patterns that resemble the cell membranes or metabolic networks found in living organisms. Their ability to form ordered structures sparks debates on the definition of life and the origins of biological complexity. This phenomenon challenges traditional boundaries between living and non-living matter. Some permafrost studies have documented molecules that spontaneously organize into vesicle-like structures, similar to primitive cell membranes. It's like watching a row of dominoes fall in a perfectly organized pattern. While the arrangement seems purposeful, it is simply the result of natural forces and design constraints. One major scientific challenge is determining whether complex molecular structures in permafrost are produced by biological activity or through purely abiotic processes. Researchers use isotopic analysis, molecular markers, and structural patterns to differentiate between life-driven and non-life-driven chemistry. This rigorous approach helps to establish whether the observed complexity is due to past microbial activity or can be explained solely by geochemical reaction. Understanding these distinctions is critical for studies in origin of life research and astrobiology. In one study, scientists compared the carbon isotope ratios in permafrost compounds to those typically found in living organisms and discovered differences that support an abiotic origin. It's like comparing fingerprints. By carefully analyzing the patterns, you can determine if they were left by a person or if they were made by an automated machine. Studying pseudo-organic life in permafrost provides clues about the potential pathways through which life might have emerged on Earth. If complex, lifelike structures can form abiotically, it suggests that the building blocks of life might arise naturally under the right conditions. This research has profound implications for our understanding of how life could originate on other planets with extreme environments. The findings challenge conventional models by demonstrating that lifelike complexity does not necessarily require biological intervention. Research into permafrost molecules has inspired hypotheses that early Earth, with its extreme conditions, might have fostered similar abiotic processes that eventually led to the first living cells. It's like discovering that a simple recipe can yield a surprisingly elaborate dish, suggesting that even without a chef's intervention, nature might have its own way of cooking up life. Advanced analytical techniques such as mass spectrometry, nuclear magnetic resonance, NMR, and electron microscopy are used to study the structure and composition of pseudo-organic molecules in permafrost. These methods allow scientists to characterize the chemical and physical properties of the molecules with high precision. Detailed analysis helps in understanding the formation mechanisms and potential functions of these complex structures. This rigorous technological approach provides the evidence needed to explore whether such molecules could serve as precursors to life. Scientists may use mass spectrometry to determine the molecular weight and structure of compounds isolated from permafrost, revealing patterns that hint at self-organization. It's like using a magnifying glass and a digital camera to capture every detail of a tiny insect. These tools help reveal the intricate beauty and complexity that would otherwise go unnoticed. The cold, stable environment of permafrost plays a crucial role in preserving complex pseudo-organic molecules over long periods. Low temperatures slow down chemical reactions that might otherwise degrade these structures, allowing them to persist and evolve. This stability provides a unique window into the chemical processes that can occur in extreme environments. Studying these conditions helps scientists understand how lifelike molecules can remain intact, even in the absence of biological processes. Samples from Siberian permafrost have revealed well-preserved molecular structures that provide insights into ancient geochemical reactions, offering clues to the prebiotic chemistry of early Earth. It's like preserving a delicate piece of art in a climate-controlled museum. The environment protects it from decay, allowing its intricate details to remain visible for generations. Research into pseudo-organic life in permafrost has significant implications for astrobiology and the search for life beyond Earth. If lifelike molecules can form abiotically in extreme terrestrial environments, similar processes might occur on other planets or moons. Future studies aim to replicate these conditions in the laboratory and compare findings with extraterrestrial samples. This line of research opens up new avenues for understanding the universality of chemical evolution and the potential for life in the cosmos. Scientists plan to simulate permafrost conditions in the lab to see if similar complex molecules can form, which could then be used as a model for detecting prebiotic chemistry on Mars or icy moons like Europa. It's like testing a recipe in your kitchen before serving it at a big dinner party. The laboratory experiments help ensure that nature's recipe for life could work elsewhere in the universe.